Worms are everywhere. In the ground, in the water, at the bottom of oceans, within the ice of Alaska, and even, sometimes, inside us. Their wriggly, slimy bodies are quite repulsive. Yet in a strange twist, they're turning out to be valuable allies.
The strength of the animal is its simplicity.
Worms have helped scientists solve great puzzles in genetics and disease. They've even helped map the human genome.
The worm contains the secrets of life.
They've given us clues to combat the parasitic diseases that afflict over 1 billion people worldwide.
Clearly, we are understanding more about how the immune system works by learning about all these parasites.
And worms quite literally hold the earth together. These bald, slimy creatures have few friends among humans, except perhaps amongst anglers. These two men with spades are not in fact body snatchers, they're worm watchers. Kevin Butt and Chris Lowe are experts in soil ecology. They often visit this cemetery in Preston, England, to study the galleries of earthworms living amongst the dead. Worms account for over 2/3 of all the creatures living underground. Their rule is supreme, so their impact is huge. A good reason for the scientists to take a close interest. But getting them out of the ground is a challenge. Kevin and Chris are using a 12-volt car battery to generate an electric field. This pushes the worms to head towards the only way out, the surface.
The ring defines an area of 0.2 of a square metre, and we base our findings on only animals that come up within the ring. Anything that comes up outside is not counted.
The car battery is one technique. There are others. Birds have come up with their own methods.
Birds tapping their feet on the ground, it's thought mimics rainfall, and this brings the worms to the surface. This particular method with the electricity going through the soil is a bit more extreme, and really they're trying to escape from something that's really quite painful to them. Here's one. This is Lumbricus terrestris. This is the largest earthworm in Britain. This is a deep burrower and one that can go down to 2, possibly 3 meters. I'll put it into the water to stop it being stressed.
All the worms that emerge within the circle are carefully put aside.
We then weigh the earthworm, so we know its mass, we know how big it is, and we can relate each earthworm back to its particular burrow.
A cast is taken of each of the worm burrows using synthetic resin. It has to be left to dry for several days before the casts can be recovered. The importance of worms as simple, small masters of the underground was first recognized by the 19th century's most famous biologist, Charles Darwin. He suspected them of causing historic ruins to be swallowed up in the earth. Along with an ingenious experiment to test his theory, he published a seminal work on the role of earthworms.
Within it, there are lots of things, some on earthworm intelligence, these days we dismiss. But other things, earthworm ecology, their actions on the soil, still regarded by many to be really, really important, can't be ignored.
Darwin proved that though earthworms have no eyes, they are nonetheless sensitive to light, that they cannot hear music but can feel the vibrations made by a piano. He then decided to prove that worms play a key role in regenerating and displacing soil. He thought of a simple experiment he could carry out at the bottom of his garden. First, he placed a millstone down on a patch of earth that was full of worms. Then he waited and waited for the worms to make the stone disappear. Darwin thought that given time, the worms were capable of shifting huge quantities of earth. He realized that when a worm digs a burrow, it actually swallows the earth in front of it and digests it by means of a special gland. He postulated that all the vegetable mold in the country, that's to say, all the soil enriched with decaying vegetable matter, had probably passed through the worm's intestines several times.
He came to the conclusion that it was his vegetable mold that was responsible for the burying of these monoliths. In other words, it was worm-worked material that was taking these things down into the ground.
Darwin's findings were not generally accepted until the 20th century, but lay the groundwork for today's worm scientists.
We come to the cemetery to look at the burrows created by Lumbricus terrestris, usually in the spring or in the autumn, because those are the best times of year, both in terms of the soil being at its best, but also in terms of earthworm activity. The advantage of working in a cemetery is that they dig holes here all the time, big holes. And actually, this is good. Take that from. Really? Try and trace it back to the top.
From the casts, Kevin and Chris will build a picture of the earthworm community. It's a way of peering into the worm's hidden world.
We often get asked questions like, do these earthworms go down 2 meters? Do they reach the graves, the dead people there? In general, earthworms are detritivores. They just take decaying organic matter, but of the vegetable type. So there's no concern. People shouldn't be worried about earthworms going down and eating their bodies when they're dead.
Each worm burrow is like a bachelor pad. Every worm has its own individual hole, which it only leaves to get in extra food or to look for sex.
It takes a mature earthworm maybe a matter of hours or perhaps days to create a burrow of this type, if it's in a new setting. But this is a very unusual occurrence. What normally happens is that a burrow, having been used by one animal, if that animal dies through natural causes or is predated, then it'll be taken over by another animal. For this reason, burrows can exist for decades, perhaps even centuries.
And this ability to tunnel, turn soil over, and mix in vegetable matter means worms may hold the solution to a human-made problem. Decades of industrial farming methods have badly depleted arable land in many areas of the world. Clearing, irrigation, the use of pesticides and fertilizers have destroyed life in the soil and stripped it of nutrients. The earth becomes powdery and is easily eroded. In some parts of the world, soil can no longer yield the crops desperately needed to feed people. Genola Perez from the University of Rennes in France is just one scientist who believes earthworms can be used to revitalize the soil.
Now we just need to shape this soil sample into a cylinder, and then we can move on to the tomograph. Why are we looking more closely at earthworms than at other organisms? Because they represent the majority of the soil's biomass. In a hectare of meadow, you'll find 1.5 tons of worms. But they are also strongly affected by environmental factors, and especially by farming techniques. And so we increasingly need to study these organisms to be able to judge the quality of our soil.
The number of worms in a soil sample, their size, and the density of their burrows are a means of gauging how healthy the soil is. Using this medical scanner, Ganola can reconstitute worm activity in this block of earth with astonishing accuracy.
Now we're looking at the block of earth we extracted earlier. More precisely, we're looking to see how porous it is. Anything that appears in black is a hole made by an earthworm.
Close to the surface, the smallest worms have built a dense network of horizontal burrows, while the larger worms have dug tunnels plunging far deeper. These wormholes play an essential role in fighting erosion, as they allow rainwater to seep further down into the earth. As the water percolates down, it carries with it the organic matter that the worms have worked into the ground, and it is so vital to plant growth.
We can see that the earthworm changes both the physical and chemical characteristics of the soil. That's why nowadays we call the earthworms soil engineers.
Ganola's work shows how dependent we are on worms for successful farming. But are there enough worms to do the job of resurrecting our farming soil? Our English wormologists are looking at how worms reproduce.
I can see one. Here. I think they're mating.
Lumbricus terrestris is one of the only earthworms that mates on the soil surface. The two animals come to the surface, keep their tails in their burrows, come together, and will stay copulated for 2 or 3 hours as they exchange sperm.
Weirdly, earthworms are hermaphrodites. They have both male and female sex organs, and both are active simultaneously. But the worms must still mate for fertilization to take place.
If for some reason they're disturbed, perhaps a predator comes along, they can rapidly retreat back into the burrows and avoid being eaten.
Elsewhere in Europe, other scientists are taking a close look at the sex lives of worms. In the botanical gardens of Tübingen in Germany, Nico Michiels and Gregor Schulte are studying the sexual behavior of worms precisely because they are hermaphrodites. The aim is to find out how these animals handle their dual sexual identity.
We have a copulating pair here.
The scientists work using red light, which the worms cannot perceive. In this way, the humans themselves go unnoticed.
The reason why we have to go out to observe earthworms at night is that they will only come to the surface when it's dark and humid. During this time, they might be interested in interacting with a neighbor. And then eventually, after a long series of mutual visits or interactions, they might decide to copulate. And this is what we see right here.
But why do they mate with another worm when they have both sexes themselves? Scientists think it produces a cross between two types of genetic inheritance, and so gives worms a better chance of adapting to their environment. Professor Michiels, an evolutionary biologist, believes that hermaphrodites have been largely neglected in evolutionary studies until now.
By studying hermaphrodites, I'm studying the ultimate equality, sexual equality system. Everybody is the same in a hermaphroditic population. Everybody has the same goals, the same preferences, want to do the same things, want to achieve the same things, has the same options, the same biological structures, everything is identical. If you generally look at hermaphrodites and you compare them to species with separate sexes, the first thing that you will notice is that the hermaphrodites have very poor eyesight, they don't fly away, they don't sing, they don't have, or usually don't have colorful patterns to show. And this is very different from insects, mammals, birds, whatever.
So earthworms apparently don't have to go to a lot of effort to seduce each other. They are unadorned because they are hermaphrodites. To study worm mating behavior in greater detail, our German scientists have set up artificial burrows in an air-conditioned room and will film what the worms get up to.
We've set the air-conditioned room in such a way that the earthworm's day-night rhythm is inverted, so that we can watch them mating in the daytime.
This peculiar voyeuristic activity confirms Nico Michiel's suspicions. There's something very odd going on between the mating worms.
From my first experience, it was very obvious that hermaphrodites had very bizarre ways of exchanging sperm and inseminating each other, that it was very obvious to me that something really strange is going on in hermaphrodites.
These photos, taken with an electron microscope, show the skin of a worm after an exciting night. It looks like it's been through a bombardment. These nasty piercings are caused by its partner, with these weapons seen in magnification, double rows of very nasty little daggers. Both the worms have these weapons, and each tries to use them on its mating partner. But why? To force the partner to take the female role and to stop it from mating with another worm.
All individuals in a population of hermaphrodites will prefer to take up the male role, at least initially. And this means that you have basically not a population of individuals of two sexes, but basically when it comes to mating, it's a population of males, where everybody's trying to inseminate everyone else, and nobody's not too much interested in playing the female role all the time.
Yet it's females who lay eggs and allow the species to continue. Nico Michiels thinks that the stubbornness of worms in all trying to act like males has locked worms into an evolutionary dead end.
This escalation has slowed down the evolution of hermaphroditic groups much more than in separate sex groups, where there is also sexual conflict, but not to the same extent. Sometimes I have the impression that we tend to believe the illusion that males and females, men and women, are identical, but obviously they are not. And I think my research shows for very good reason, if we all would be the same, if we really would be the same in a sexual sense, then we would, without any doubt, end up in the same kind of ridiculous sexual escalations that we see in hermaphrodites. And from my perspective, this is the reason why hermaphrodites have been stuck in this worm stage rather than evolving further on.
Yet it is precisely because the worm is a hermaphrodite that it was suddenly propelled to the forefront of biological research. It was to play a vital role in the biological revolution of the 20th century, the mapping of the genome. The worm owes its success to a remarkable biologist from the Laboratory of Molecular Biology at Cambridge University, Sidney Brenner. This laboratory has produced many Nobel Prize winners, including Francis Crick and James Watson, who received the award in 1962 for their discovery of the structure of DNA, the double helix which carries our genetic blueprint. When Brenner was a young scientist, it was already known that genes were responsible for transmitting certain characteristics from one generation to another. It was also known that they were involved in cell development and that their instructions were relayed through proteins. Most of these discoveries had been made through a study of single-celled organisms such as bacteria. Sidney Brenner believed the next step was to look at how genes control the development of more complex organisms.
Although we understand now how genes make molecules, we don't know how genes make hands. I mean, many people have said, why aren't we doing brain research like many other people? Why don't we try and work on how the brain works? That, of course, I think is the important challenge of science. But I think we would like first to try and understand how the brain is built.
But Brenner needed a laboratory animal to work on. He started with the fruit fly, already familiar to geneticists. But for Brenner, it didn't fit the bill. Its legs, wings, and eyes made it too complex. So he looked for a simpler animal, something whose behavior and anatomy would make it easier to study. He started to consider using small worms. He looked at 60 species of worms from the group known as the nematodes, which in their natural state live in the earth. Finally, he settled on an English worm barely a millimeter long, but with a pretty impressive sounding name, Caenorhabditis elegans, soon shortened to C. elegans. From a practical point of view, C. elegans worms have many advantages. They're very easy to breed, don't take up much room, and are cheap. They eat only bacteria and can survive perfectly well in little plastic boxes or Petri dishes. Between experiments, they can just be put back in the fridge. Project C. elegans started in 1965. This little worm was to become famous, for it had one particularly valuable quality, as LMB geneticist Mario De Bono relates.
The strength of the animal is its simplicity. And the criticism was that it was too simple, but actually the simplicity is also a big advantage because you can then understand at the level of individual cells, moreover at the subcellular level, and individual molecules. And that's something that is rather harder to do in other model organisms.
Simplicity is not the only quality of this hermaphrodite worm. It's able to fertilize itself and give birth to a large number of offspring. And the young reach maturity in just a few days. The mere 1,000 cells which compose the worm's body are visible through its transparent skin, which makes studying it much easier. Brenner and his team set out to make mutant worms, whose genes could be manipulated to show their impact on the worm's development and functions. Over several years, the worm's genes were methodically broken down into their component parts. Using chemicals and radiation, the scientists modified the genes which the worms passed on to their offspring. In this way, the scientists could create whole new generations of worms with specific mutations. In this great cause, millions of tiny worms were sacrificed.
So the early mutants that were identified were really very, very poorly. They were very sick, they were very uncoordinated. And what that meant is that they were very obvious. You could spot them a mile away that something was wrong with them.
The biochemists carried out extensive experiments on the worm's chromosomes, and these manipulations gave birth to all sorts of weird creatures. Dumpy here gets his name from his abnormally short, squat shape. Roller is stuck in a bent position and has a tendency to wriggle round in circles. When geneticist John Sulston joined Brenner's Project C team, he knew nothing about these tiny worms. but they seemed to him perfectly suited to meet Sidney Brenner's goal, to understand how DNA pilots cell development.
The central thing is mutants that allow you to pinpoint some defect in the animal. Then you can go back into the genome, find the defective gene, and then you say, ah, this gene now is used in this process, and then we can figure out what's going on.
Sidney Brenner made a meticulous record of how mutant genes affected the worms. After 8 years of hard work, he had managed to identify 550 of C. elegans' 20,000 genes. This was the first step towards mapping a genome. At the same time, the worm's anatomy was also undergoing minute investigation. Using a machine with a diamond cutting blade, it was possible to slice cross-sections of worm less than half a micron thick. In order to get a complete picture of a worm 1 millimeter long, 13,000 slices were needed. Then technician Nicol Thompson took photos of these thousands of slices using an electron microscope. They were key to understanding how the mutations had changed the composition of cells in the mutant worms. Thompson's achievement deserves an entry in the Guinness Book of Records.
The key thing about this is not just one section, but the fact that Nicol could produce hundreds or thousands of sections without losing any.
Yeah, that was an important thing.
And this meant that one could trace through and discover in 3 dimensions the complete nervous system. And you can see the cuticle, the ale on the outside, you see the pharynx in the middle, a feeding organ. But very particularly is this region labeled nerve ring here. And that's very important because this is it's like the brain of the worm. That's where the thing thinks whether to go forwards, backwards, and when to eat and so on.
John Sulston was soon to focus all his attention on the C. elegans nervous system. For a biologist of the time, there was no finer challenge. Before studying the complexities of the brain, it was necessary to understand how it was constructed, how it was wired up. When Sulston started on this work, the generally held belief was that when the worm hatched from its egg, its nervous system was already fully formed.
I noticed that some of these cells were absent when the worm was first hatched. When you look at this diagram, you can see the eggs of the worm here, and then they hatch and give these little tiny larvae. Those cells were not there at that stage, but they were there at that stage. So I wanted to know where they'd come from.
To solve this mystery, Sulston planned to observe the growth of the nerve cells and to try to understand how these cells related to each other from the very moment the worm hatched. The worm's transparent skin was a great advantage. However, keeping the wriggling worms within the viewing field of the microscope was a great challenge.
To give it food, that was the key thing. Everybody thinks, oh, you put the worm under the microscope, doesn't matter if it dies, just stick it there. And I realized, for some reason, I thought, oh, it wants something to eat, and it was. It immediately slowed down. Now it was just moving, and I could see the cells.
This deal turned out to be very profitable both for Sulston and for the worm. By giving the worm what it wanted, he was able to become the first biologist to watch a living being grow cell by cell.
And it was a very exciting day for me when I first saw a cell division. For the first time, I realized I was going to be able to trace the cell lineage.
From that point, Sulston was bent over his microscope day and night, following the growth of each of C. elegans cells from birth to maturity. Once he'd identified a cell, he didn't let it out of his sight. He took endless notes in a kind of graphic shorthand, setting out each division and displacement of the cell minute by minute.
I remember saying to John at the beginning of this, I said, Johnny, it's going to take me 1.5 years to do this. Is it worth it? And he said, yeah, of course it is. So I said, all right. And here you see the result of that. Eventually, I was able to follow the division of a whole set of precursor cells here through a repetitive pattern. Each one divides in more or less the same way to produce a set of daughter cells.
For the first time in the history of biology, the cell lineage of a living being had been mapped out in its entirety, providing geneticists with a precious tool for future research. In the course of this task, John Sulston noted an unusual phenomenon. A certain number of cells, 131 to be precise, were born and died without dividing.
I just realized that cells were disappearing. And I realized that actually they were disappearing not sort of gradually, but in a very distinctive manner, so that they actually became sort of glistening in the Namaski, so I could recognize them.
The fact that 131 cells inevitably died during the embryo's development implied that this was no matter of chance. Sulston suggested it was due to a genetic program that governed the life and death of the cells, and he was right. The same phenomenon occurs during the growth of a human embryo. For example, hands are webbed at first, before the cells of the webbing die, allowing fingers to appear. This programmed cell death is a key part of the development of all living beings. Discovering the role certain genes play in determining whether cells live or die was to lead to far-reaching medical advances.
These genes turn out, in a number of cases, to be important in the human body. And we find that the homologous genes in our cells can be involved in for example, neurodegeneration, where cell death is started inappropriately, can be involved in cancer, where you don't have cell death when you ought to, so the cancer grows, because it's no longer subject to that control.
When a C. elegans genome is compared with a human genome, it can be seen that worms and people share about half the same genes. So scientists can experiment on worm genes to study the cell mechanisms involved in human diseases like cancer, Alzheimer's, or Parkinson's. Thanks to C. elegans, we can also safely test gene therapies designed to repair disturbances to a cell's normal life.
What we've learned from this molecular revolution that's been going on since the early 80s is that we can learn most quickly about ourselves, a lot of information, by studying simpler organisms like C. elegans.
In 2002, Sidney Brenner's team was awarded the Nobel Prize for Physiology and Medicine for their discoveries on how genes regulate the development of organs and on programmed cell death.
Dear Dr. Sidney Brenner, I wish also to win a Nobel Prize. Please tell me how to do it. You need to discover the right animal to work on. For example, you could try a worm. A worm with possibly a name like C. elegans.
A superworm. The Nobel Prize-winning worms are still in the lab, carefully preserved in bottles of liquid nitrogen at minus 196 degrees Celsius. They constitute a real reference source of cryogenized organisms. And C. elegans worms have an astonishing quality. They can come back to life after lying dormant for years.
So if we wanted to do the experiments all over again, we can, because all the worms are still here, frozen away. And we can bring them back to life by simply taking them out of our liquid nitrogen, taking out the worms, putting them onto a plate, and in 15 minutes, the worms will start crawling again. And we still send these worms all over the world, wherever C. elegans research is being done, and even into space.
In January 2003, hundreds of thousands of worms were launched into space on board the ill-fated Columbia Space Shuttle. The worms were to be used in research for future manned trips to Mars. Once more, the worms were to serve as a model for humans, giving precious information about the effects of long-term spaceflight on body tissues.
The ultimate goal was to study problems that will occur during very long voyages, the effect on muscles, nerves, certain types of behavior, and on cells. Particularly, given the level of radiation astronauts will be subjected to on a voyage to Mars.
First, the scientists had to set up the right conditions for the worms to survive on the shuttle.
So the basic goal was to use a different media for growing worms because this media would make experimentation in space easier. A lot of times astronauts don't have time to tend to experiments, so if we could design something where the worms don't require attention, and if we could grow them in liquid and actually automate the experiment using pumps, then you don't have to use astronauts.
After 17 days in Earth orbit, the mission reached its end. The worms seemed to thrive in their liquid environment, and many experiments had been successfully completed. The shuttle began its return to Earth.
As you know, the shuttle broke up as it was coming back to Earth. And work from the Columbia Accident Investigation Board shows that the shuttle was traveling in about Mach 19 and was about 61 kilometers above the Earth when it started to break up.
The aluminum canisters containing the worms were thrown out of the craft at an altitude of about 40 kilometers. At that point, the shuttle was still traveling at 800 kilometers an hour.
After the breakup, the big concern that everybody had was, well, did anything survive? Did anybody survive?
It soon became clear that there would be no survivors. But 1 week after the crash, one of the first aluminum canisters was found in Texas. Then, one after the other, the rest were recovered. Nearly 3 months after the accident, the scientists were given permission to open the containers. The first surprise was that the Petri dishes within had survived the intense heat generated during their fall to Earth. After studying the dishes a few minutes, the scientists had a second surprise. Miraculously, the worms were still alive.
The two plates that had all the worms that we would have expected to actually be there were plates that had run out of food in space as predicted because they were the NGM plates, and those animals were in the dour state, so they're non-reproductive state.
This miracle could only occur because faced with a lack of food, the very young worms were able to put their development on hold. This is what is known as the dour state, a kind of self-induced lethargy.
What they do there is molt into a rather shrunken-looking worm with an altered cuticle that's specialized, an altered skin which is specialized for them to resist desiccation and other environmental insults.
Worms can not only survive for several months in this state, but are also capable of returning to an ordinary worm life.
The dour animals tend to be somewhat more resistant to stress, and so one theory is that the worms actually, in that case, survived because they were able to tolerate the heat a little better than the reproductive animals.
Worms can be very resilient. This ability to alter their metabolism is just one of their survival skills. In fact, worms can survive in the most unlikely places, even inside the human body. Worldwide, over 2 billion humans are infested with parasitic worms.
A successful parasite makes sure that the host lives as long as possible, and they gain the maximum amount of time to reproduce and to continue to contaminate the environment.
Parasitic worms produce extraordinarily large numbers of offspring. Some produce as many as 200,000 eggs a day. They need this huge number of offspring to ensure their survival, as the journey from egg to adult worm is long and hazardous. As soon as a larva hatches, the countdown begins. It must find its first host within 2 days or die. Once installed in the first host, it will wait, sometimes for months, for a more permanent host to come along. Then someone will step in water and be infected that way. Or a fly will deposit the parasite on someone's skin. Or it'll find its way into a human by means of a glass of water. A parasite that makes this second stage settles into the human body for a long stay. The next task will be to find a way to release its own eggs in a favorable environment so that the cycle may continue.
Even if you were a science fiction writer, you could not come up with something so, so amazing. You know, it's really quite fascinating. It is quite fascinating.
After millions of years of evolution, parasitic worms have become extremely efficient. They can remain for a long time hidden within a human body, up to 20 years.
Clearly, most of the problem with these worms is that you live for a long time with them. And therefore, it's the debilitation, the lethargy, that is more of an issue because then you cannot be as productive.
Parasitic worms cause serious diseases, which devastate entire communities. In some African villages, for example, the majority of adults are struck by blindness due to the larvae of certain threadworms. As a result, the whole village can be brought to ruin. Over the past decades, the World Health Organization has been waging a sustained war against parasitic worms, which afflict some of the world's poorest countries. Campaigns have been launched to target the larvae and the aquatic organisms which enable some worms to spread. Vast reservoirs of water are treated with chemicals to stop the parasites reaching the human population. In some cases, it's worked, and certain worms have been almost entirely eliminated. Unfortunately, others are managing to resist. One of the most sinister of these is the schistosome. Schistosomes use snails as an initial host. No chemical can kill the snail without also killing the local fauna, so the parasites thrive in the first host and spread effortlessly to the second, humans. Here they've developed a lethal advantage.
Schistosomes are almost perfect parasites in that they live in the blood system of humans where they should be attacked by the immune system, but they do not.
The schistosome can settle in the human body for long periods because it somehow eludes the surveillance of the body's immune system.
The schistosomes may be mimicking host functions so that they appear invisible to the immune system.
But how does the parasite manipulate its host in order to ensure its own survival? Frédéric Thomas, an expert on parasites, has studied the biological mechanisms of a nematomorph worm known as Paragordius tricuspitadus. These worms are rarely seen, as they're hidden within their insect hosts, usually a particular type of cricket. They only emerge when it is time for them to reproduce, which they have to do in water. Thomas decided to use this worm as a model to research how parasites function.
Most parasites are small compared to their hosts. Here it's easy to tell that we're dealing with a macroparasite, a very large parasite. Keeping relative proportions, it's like a human having a watering hose 8 to 10 meters long in the stomach. It's a gigantic parasite that has grown from being a microscopic cyst to a very large worm without damaging the cricket's internal organs, which it needs to keep alive until the end. And so it's only at the very end that it will change its host's behavior, forcing it basically to commit suicide by jumping into the water.
Thomas has studied these parasite-dictated suicides in what may seem a surprising setting, swimming pools in the south of France. On some balmy summer evenings, he has recorded as many as 200 of these suicide attempts by crickets.
All species of nematomorph worms need water to reproduce. They're parasites which as adults are necessarily free to move about, and more importantly are aquatic. So once they infect a terrestrial insect, they're obviously faced with a problem, which is how to get back to the water.
Before working its way out of the cricket's abdomen, the worm must first use all of its manipulative skills to get the insect into the pool. After all, a cricket's not normally an avid swimmer.
In fact, if the cricket goes looking for water, it's not its own genes telling it what to do, but actually the genes of the parasite.
How can a worm, little more than a simple tube, possibly take control of an insect? Thomas used the most sophisticated tools of molecular biology to find out. He discovered that an infected cricket's brain produces twice as many neurons as that of a healthy cricket. But this was not all.
One of our most surprising discoveries was the existence of apparently mimetic proteins, proteins that are produced by the worm, but which are structurally similar to those we know to belong to the insects.
The worm has the ability to copy the cricket's molecules, modify them, and then send them back to the cricket. Using these forged molecules, the worm fools its host. To begin with, it makes the cricket behave erratically, leaving its usual natural habitat. The parasite then goes on to push the cricket to head towards water. This ability to send molecular messages is not exclusive to this particular worm. Scientists have detected the same phenomenon in other parasites.
Our work on the nematomorphs has made it possible for us to save a considerable amount of time on other areas of research. Research we're carrying out in connection with public health threats such as malaria or sleeping sickness transmitted by the tsetse fly and mosquitoes.
Understanding the worm's strategies makes it possible to understand how other parasites manipulate their hosts. The most deadly of them, the malaria parasite, kills a child every 30 seconds in Africa. The parasite is not a worm, but it operates in the same way. It modifies the behavior of its host mosquito, pushing it to bite the victim which suits the parasite best. Once it is installed in its definitive host, man, it is thought that the parasite causes the human to give off a substance which attracts other mosquitoes. In this way, the parasite can easily be transported to other victims.
Knowing what the parasite's strategies are is the first step in being able to invent countermeasures.
Worms are amongst the oldest creatures on Earth. Slimy, wriggly, ugly, and loathed, it turns out that these ancient rulers of the underground have much to offer us. Now scientists are using them to improve understanding of human biology.
The worm, even though it only has 1,000 cells and even though it's only 1 millimeter long, contains the secrets of life.
Its capacity to regenerate our soil has earned even the most common of worms, the earthworm, a new respect. It does appear, indeed, that the worm has finally turned.
If worm in the soil has made it up to this point where we can observe it, they obviously have been equally successful to ourselves.